That's C1.
Yay! Somebody did it.
Okay, so it is recording? Okay, great. Okay. And so that's C1. And the carbon that's right next to it is called the alpha carbon. Yes, this meeting is being recorded. Okay, great. The one next to that is beta. Next to that is gamma. And whatever is at the very end, at the very opposite end from C1, that will be the omega. Okay, that's what this Greek letter here is. Looks like a W. That's omega. In the case of palmitic acid, that's C16. Okay? So the two kinds of fatty acids, they're saturated versus unsaturated. Saturated fatty acids is shown here on the top, and that means there are no double bonds, right? Like this. And you hear maybe in the popular literature and advertisements or from the doctor, saturated fatty acids, you kind of negatively refer to those because those increase your risk for a lot of cardiovascular diseases and other, disease-oriented risk factors. Unsaturated fatty acids means that there's a double bond present somewhere along this carbon chain. So, and when you hear omega fatty acid, like omega-6 fatty acid, that means that 6 away from the omega carbon, so 1, 2, 3, 4, 5, 6 carbon away from the omega carbon, that is where the double bond appears, right? And you hear this, you know that they're omega-6, omega-3 fatty acids. And so you see the two essential ones. There are only 2 essential fatty acids, meaning that we don't make it ourselves. We must acquire it from foods listed here at the below as a source of omega-6, omega-3 fatty acid. Linoleic acid is shown at the top. And the other one that's essential is alpha-linolenic acid, which is shown here at the bottom. These are important because they're precursors of arachidonic acid, which is another important pathway in the body, which we're not going to go into here. But so these are the 2 essential fatty acids. And so what's the difference? Why is saturated fatty acid not so good for us? Well, you see here, the saturated fatty acid has strong intermolecular interactions, because this is all very hydrophobic and very orderly, symmetric. And so they can really form a solid. It has a higher melting point, right? Because it's saturated, it's solid, like you see it in cheese, hard cheese, butter, milk, meat. They are rich in saturated fatty acids. And so they're less readily oxidized because of this property and increases the risk for heart disease. Whereas unsaturated fatty acids like oleic acid, which is rich in olive oil, there are weaker intermolecular bonds between the different fatty acids. And so they don't pack as readily as a saturated fatty acid, and they're more readily oxidized for ATP production, which is the whole purpose in terms of we think, what's the benefit to us? That is the benefit of fatty acids, so that we can obtain the energy currency from those molecules. And so how do we get triglycerides? We get it from the diet. So when you take in a meal that has fats in it, the triglycerides, they're lipids, all right? And so they're hydrophobic. And so they mix in the gut lumen with bile. Bile is a substance, it's kind of like a detergent that emulsifies the triglycerides into these droplets, like shown here. And why that's important is that now those emulsified droplets of fat can be accessible to pancreatic lipase. All right, lipase is an enzyme that will break down a certain kind of lipid. So pancreatic lipase comes from the pancreas, therefore it gets its name. That when you eat something, the pancreas gets stimulated, it releases lipase into the gut so that now you can break down those triglycerides into their constituent subgroups. Free fatty acids, those chains off the glycerol backbone, and monoglycerides, the glycerol backbone. And so it'll get broken down. So the purpose is so that now these, they're called micelles because they're surrounded by, an emulsification here that makes it possible to cross the small intestine brush border. That's the whole purpose of this pancreatic lipase, is to make it possible that the small intestine, which is the absorptive organ in our body, can take it up. But the main purpose is just to get it into the small intestine, because once it's in the small intestine, what ends up happening is now the triglyceride is going to reform again. And you'll see this in a couple of cases in our discussion this evening, is that certain molecules will get converted into a form, let's say broken into certain pieces, to cross a membrane. And once they've crossed the membrane, they're going to get reconstituted. So that's what's happening here. So you reform the triglycerides here in the gut, and they get packaged into a lipoprotein particle, which is called a chylomicron. So in the next lecture, we'll talk all about these lipoprotein particles. LDL, HDL, maybe you've heard of those because it's a lot in advertisements for, cholesterol lowering disease, the good cholesterol versus the bad cholesterol. Chylomicrons are one form of a bad cholesterol because they're really transporting fat. A lot of triglycerides, the more triglycerides you eat, the more chylomicrons you're going to make. And so in terms of triglycerides and how they, operate in the body, they get stored in adipose tissue, okay? And they wait there until they're called into action. And so how they get mobilized, out of adipose tissue is that there are hormones. Hormones, glucagon, cortisol, epinephrine. Maybe those sound familiar to you from, high school. And those particular hormones will stimulate the breakdown of the triglycerides into fatty acids so that the fatty acids can be released into the bloodstream and then go to organs where they're needed for energy. So for example, you have a hormone, which is shown here in the blue dot. It will bind to a receptor on the adipose tissue membrane, okay? And when it binds, it will activate a whole signaling cascade. And you needn't know the details of it, but just know that when the hormone binds, now there's going to be a signal transduction, a whole signaling system within the cell. And it's going to rely on certain forms of ATP called cyclic AMP. Okay, that's different than ATP or ADP. It's a cyclized form of AMP, and that will activate a kinase in the cell. Yeah? And that kinase is called protein kinase A. Yeah, you get the gestalt of this. All the details are not so important if you're not so familiar with these pathways, but this is how the cell knows what to do. Because when this kinase, pKa, gets activated, it's going to phosphorylate. It's going to add a phosphate group to another lipase, a hormone-sensitive lipase. And when you hear the word lipase, that means that it's going to break down triglycerides. Triglycerides will get broken down by this hormone-sensitive lipase into free fatty acids. And once that happens, now these free fatty acids can be released from the adipose tissue, and then they can circulate throughout the body. So if you have a medium chain or a long chain, then you have to bind to a protein in order to get around in the blood, okay? And because they're hydrophobic, they don't really mix with the aqueous extracellular space, the serum or the blood. And so they will bind to a protein which is called albumin. Albumin is the most abundant protein in our blood. So it's there for this purpose, so that it can bind, for example, to triglycerides and ferry them throughout the body. So once you get the fatty acid to a particular organ, how do we get the energy we need from it? And that whole process is called fatty acid oxidation or beta oxidation, because it's referring to that beta carbon, okay? And this whole process of oxidizing fatty acids is occurring in mitochondria, Okay, so that's the central hub, the central place where ATP generation occurs. And so there are a number of steps. So the fatty acid is going to be taken up, across the membrane of a particular organ. And so it will go into the cytosol. The long-chain fatty acids get taken up from the blood by a cell. And in the cytosol of the cell, they get acylated. Okay, they get modified to form a fatty acyl-CoA. And that reaction requires ATP. So you just have to know that the long-chain fatty acid needs to be modified into a form which is called fatty acyl-CoA. And that is the form that can go into mitochondria. Unless that happens, we can't get, the ATP generating energy from the fatty acid. And so this is going to happen kind of sparingly because you see this process requires ATP. And if you see that a particular reaction requires ATP, you know that that's going to be done only when you really need to do it. Why waste ATP on something that you don't need to do? So when you really need to extract, what the fatty acids have in terms of generating ATP, then you'll undertake this process to make the fatty acyl-CoA. Okay, so this looks complicated, but you just follow the numbers here. You see it. And it's also shown in this book called Lippincott Illustrated Reviews of Biochemistry. It's available in the library and also online. But if you just follow the numbers, you see here, number 1, this is where the long-chain fatty acyl-CoA gets generated in the cytosol. Okay, so this will be true for long-chain fatty acids. And so that particular form, fatty acyl-CoA, can get across the mitochondrial outer membrane through a pore. It's called poring. Certain molecules that are 6 Daltons can easily traverse the mitochondrial outer membrane and go into the intermembrane space. And so when that fatty acyl-CoA is in the intermembrane space, now it'll mix with carnitine. And there's an important enzyme on the outer membrane, which is the rate-limiting enzyme in this whole fatty acid oxidation process. And it's going to form fatty acylcarnitine, which is shown here, number 3. A rate-limiting step, it's important. Why? What does it mean to be rate-limiting? Exactly. So he's saying it's the slowest reaction in a whole series of reactions. Also, if it's rate limiting, oftentimes they are targeted for therapeutics. Were you going to add to that? Yeah, same sort of thing you were thinking too, right? And you'll see that. We'll come back to these rate limiting enzymes that, become the targets for these blockbuster drugs because they really regulate the whole process. Okay, so you have this fatty acylcarnitine, and the whole purpose of this is so that it can get across the mitochondrial inner membrane and get into the matrix. And in the matrix is where the fatty acid oxidation is going to occur. And so here again, this is another one of these membrane switch business. Thank you. Have a good evening. Yeah, take care. Yeah. So that once this fatty acylcarnitine gets into the matrix, now this other related enzyme, CPT2, is going to take the carnitine off in order to regenerate this fatty acyl-CoA, just so that now it can be utilized for beta oxidation, fatty acid oxidation, and the generation of ATP. So these are various steps. Some people are really into the biochemistry. That's not the purpose for me showing it to you here. The purpose is really that you need to know these 2 things, is that beta oxidation of fatty acids produces 3 important molecules, FADH2, NADH, and acetyl-CoA. And probably you should know that, right? Because those 3 molecules are important for what? What's acetyl-CoA? Scream it out. Yes? Krebs cycle, right? Acetyl-CoA feeds into the Krebs cycle. Yes, and FADH2 and NADH are energy carriers that feed into complex 1, NADH, and complex 2, FADH2. So from one round of beta oxidation, you're going to generate these three, reducing equivalents and then acetyl-CoA. So that if you wanted to look and see the tally of all of this, you keep, doing this beta oxidation, meaning at this carbon, beta carbon from the carbon 1 where the carboxyl group is. And at each round, you're going to get an FADH2, an NADH, and acetyl-CoA. So from palmitic acid, which is the most abundant triglyceride in our bodies, you could, in principle, generate, they're saying 131 ATP molecules, right? So there's a lot of ATP to be had from one fatty acid molecule. So those are fatty acids in the generation of ATP. What about this ketone body business, ketogenesis? This takes place only in the liver. So what you need for ketogenesis is that you have to have a lot of fatty acid oxidation, the generation of acetyl-CoA. All right. So if you have an abundance of acetyl-CoA, let's say the TCA cycle can't keep up with the amount of fats you're oxidizing, then it will funnel it toward ketogenesis. So two acetyl-CoAs will just get converted into ketone bodies, which are shown here at the bottom in blue. Yeah. It's acetoacetate, beta hydroxybutyrate, and acetone. It's called a ketone body, but it's not used significantly for energy production. Why are they called ketone bodies? Because they're just chemicals like this? I have no idea. Tried to find out, but have not really solved that mystery. And what's important here is there's a rate limiting step in converting acetoacetyl-CoA in order to make HMG-CoA.
Can you hear me now?
There's an HMG-CoA synthase.
Hello?
Hello. And so this is an important set of reactions. But if you make too much ketone bodies, right, you can see acetoacetate, hydroxybutyrate. Another way of saying is acetoacetic acid, beta hydroxybutyric acid. These are acids. And so if you make too much of these, this can lead to a case where it's called ketoacidosis, right? There's too much acid in the blood. If there's too much acid someplace, that can decrease the blood pH, which can be life-threatening. So for individuals who have type 1 diabetes, where you really need to, balance how much glucose you have available. If you don't have enough glucose available, you will start making and using ketone bodies. Oftentimes, you may smell a certain kind of odor, aroma, from the breath of somebody, and that will come from the acetone. Smelling a little bit like nail polish remover, which has acetone in it, and that is coming from the ketone bodies. And so you have to be careful about, how much ketone bodies are accumulating in your body, which can occur. If you've been fasting, you haven't eaten for a long time, you'll have higher ketone bodies. It may not be dangerous or life-threatening, but that's how your body's working in order to cover for the absence of getting fuel from carbohydrates or triglycerides. And so once you made the ketone bodies in the liver, they easily go into the bloodstream because they're these small molecules, and they can easily pass the blood-brain barrier. So if you haven't been eating a lot, and your brain, neurons in particularly, they must have glucose. They cannot get fueled off of fatty acids. They can use ketone bodies somewhat. They don't store glycogen, and so, glucose and ketone bodies are important for neuronal function. And so how do you get energy from the ketone bodies is that the reaction goes in reverse. So you can take the beta hydroxybutyrate, the acetoacetate, In this conversion here, there's an enzyme. You generate an NADH, right? So that's a carrier in the electron transport chain. You can convert the acetoacetate into acetoacetyl-CoA. And the enzyme that does this is not in the liver. Okay, that makes sense because the liver is making these, so we don't want to just, utilize something that you're trying to produce for the rest of the body. And so that will convert it into the acetyl-CoA, which then can go into the TCA cycle. So this is the reverse pathway where ketone bodies are now a source of acetyl-CoA in non-hepatic tissues. And so the synthesis of fatty acid takes place in the cytosol, right? Fatty acid synthesis, and there's something which is called the citrate shuttle. And it requires acetyl-CoA in order to make a fatty acid. But maybe you know already that acetyl-CoA is located inside mitochondria. That's where it is made. And so how does acetyl-CoA, which is a molecule that doesn't easily pass across the mitochondrial inner membrane, how does it get from the matrix and into the cytosol? And so the way in which it does this is through this process called the citrate shuttle. And the main purpose of this is to move acetyl-CoA into the cytosol. Because maybe you know this, is that the reason why we can generate ATP in mitochondria is that that mitochondrial inner membrane is really tight. You cannot move protons, right? You cannot move solutes across the mitochondrial inner membrane very easily. And that's why the oxidative phosphorylation system can work. And so there has to be another way in which you get the acetyl-CoA, which gets generated in the mitochondria, to get across the mitochondrial inner membrane. And how that happens is that the acetyl-CoA can get converted to citrate. Okay? And it does this by using another intermediate in the TCA cycle. You don't need to memorize all those details. You just know that acetyl-CoA, if you want to know it, it's oxaloacetate, can combine with acetyl-CoA using an enzyme called citrate synthase in order to generate citrate. And for this particular molecule, there's a carrier protein that can now transport citrate across the inner membrane, and then it can get across the outer membrane into the cytosol. And once it's in the cytosol, now that citrate can be converted back into acetyl-CoA. And I think it's this kind of spatial hurdle that must be met that really controls this whole process. You don't do this unless you really have a lot of acetyl-CoA around in order to make fatty acids, right? Because you'll see that this is an ATP-dependent process in order to do this conversion here, and also for fatty acid synthesis, as you'll see in a moment. It's an ATP requiring process. So you don't want to do this unless you really have a lot of ATP around. I think that's an important thing to think about. Like, at least for my lectures, I'm only interested more so in how things are regulated, And so if you just think about the energy status of the cell of your body, do you have a lot of ATP to spend? Then you'll probably spend it on these kind of synthetic pathways that require ATP. If you don't have much ATP around, you'd probably want to downregulate those things, that cost a lot of ATP. So then you can kind of predict, what's going to go on, whether you're a PhD student doing basic research, or whether you're a doctor, or you're a technician, or just in everyday life. You think about, okay, what's my body doing now? You just eaten a big meal and you've got a lot of fatty acids to, supply, oxidation. You can kind of predict what's going on. So another rate limiting step is this acetyl-CoA carboxylase, ACC. It's catalyzing the rate limiting step in fatty acid biosynthesis, and it converts acetyl-CoA to malonyl-CoA. Okay, and what you'll see in a moment is that these two, acetyl-CoA and malonyl-CoA, are the building blocks for triglyceride synthesis. And this is an ATP requiring step. And so for many ATP requiring steps, there will be regulation. And this is a major one. There are three ways in which you can regulate ACC. One is by allosteric regulation. Do we know what that is, allosteric? Something binding to the enzyme, but not the active site. So it's an other site. That's what allosteric means. It's not at the catalytic site, but at another site of the enzyme. So there's allosteric regulation. There's also phosphorylation. Phosphorylation, a modifying kinase comes in, and you'll see that the enzyme gets, turned on, turned off, and hormonal regulation. So here's the allosteric part. So when the acetyl-CoA carboxylase, okay, it's a protein, it's an enzyme, when it gets synthesized in the cell, it forms these dimers, these two balls, two dimer means two, and a homodimer, and it's inactive. But if you have a lot of citrate around, right, we need citrate in order to start the fatty acid synthesis cycle, then that will bind to ACC, not at its active site, but another site, and it will now cause the polymerization, the assembly of this acetyl-CoA carboxylase, and form this active polymer. If you have a lot of long-chain fatty acids around, which is the end product of this whole fatty acid synthesis business, that's going to inhibit this from occurring, right? Because if you have a lot of long-chain fatty acids around, why make more? So that there's a regulatory system that can dampen this. So if you have a lot of citrate, ACC activity is going to go up. If you have a lot of long-chain fatty acids, the end product, ACC activity is going to go down. And then there's phosphorylation, right? Phosphorylation is mediated by an AMP, activated protein kinase, all right? So AMP comes from ATP, which got hydrolyzed, used, and then formed ADP. That will get used and now form ANP, ATP triphosphate, ADP diphosphate, AMP monophosphate. And this AMP is going to activate a kinase. So only when ATP levels are low, right, ATP has been depleted to form ANP, now that kinase is going to be active. All right, so it knows, not so much ATP in the cell, it's going to phosphorylate this polymer of ACC, the active form, and when it phosphorylates it, it's going to turn it off, right? ATP level is low, right? AMP level is high. If ATP is low, then we don't need to do this fatty acid synthesis, spend all the ATP. So this kinase is going to respond to that, sense that, phosphorylate ACC, and downregulate it, basically turn it down, tune it down. And hormones, right? This makes sense. So if you have insulin, insulin levels are high, more glucose coming into the cell, right? More glucose, more glycolysis, more what? You're going to get more ATP because the glycolysis is going to generate one round, two pyruvates. Those will go into the mitochondria to make acetyl-CoA, right? So that insulin is going to really now allow you to make more fatty acids. So you could predict, more ATP, well, then this AMPK is not going to be working, right? There's another phosphatase that's going to sense, ATP levels are high. Now we can take the phosphate group off inactive ACC and turn it on so that now we can make fatty acids. And the reverse is true for glucagon. Glucagon is when you need glucose, right? Because it's in short supply. And so that's going to be able to communicate that we need to turn down all these ATP-dependent processes. And so that's going to favor now the inactive form, phosphorylated form of ACC, just so we can, put ATP in reserve, have it. Don't waste it on making fatty acids when we don't need it. And so in the end, the way in which palmitic acid, which is the most abundant, gets made is by this enzyme, which is called fatty acid synthase, FAS, fatty acid synthase. And what's just shown here is it's a fascinating enzyme. It's got 9 different enzyme activities, but it's not regulated. And so this just shows you is that this fatty acid synthesis, it's a big complex, and it covalently attaches with the acetyl-CoA, which is one precursor. It also covalently binds to the malonyl-CoA, which is the other building block of fatty acids. And so this malonyl-CoA is going to get conjugated to the acetyl-CoA. And it will keep doing that. It'll keep transferring. So this had only 2 carbons here. Once this malonyl-CoA comes here, now there are 4. Another malonyl-CoA will link on here. Now there will be 6. Keeps on doing this until you finally get, the 16 carbon triglyceride, the fatty acid, which generated here. So that's basically, the story of how we synthesize fatty acids, how we oxidize them, what is the determinants of whether it goes one way or the other way. When do we choose to make fatty acids? When do we turn it off? That's sort of, I think, laid out a little bit here. If you can just think about, how much ATP you have in your bodies and yourself, you'll be able to predict that. And so this is another kind of regulatory thing, is that if you had a lot of malonyl-CoA in your cell, in the cytosol, that's going to inhibit this carnitine palmitoyl transferase, right? This is for beta oxidation, right? If you've got a lot of malonyl-CoA around, and this must mean because you have a lot of ATP, this is the way that the cell knows, whether to keep fatty acid oxidation going or not. Because if you have a lot of ATP to make fatty acids, why do you need to oxidize it in order to generate more ATP? Because you have enough ATP to spend. So it's just those kind of things to think about, in terms of, how these pathways are regulated one with the other. And this I thought I would add in because I think it's really interesting, because I never really understood it before, is how these things are all integrated with one another. And I think this is a good example of how these kind of energy producing pathways, for example, involved in fatty acid synthesis or oxidation, how that can also relate to alcohol metabolism. And so, I think nowadays, amongst many younger generation, the awareness that ethanol consumption is not such a healthy thing, but you can understand that biochemically and medically. And I think this really makes a point, is that excessive alcohol consumption causes dysregulation of multiple pathways in our body. And they're listed here, but I'll show it to you. It's a little bit easier to look at it in this way, is that if you're ingesting ethanol in drinks or whatever, ethanol gets converted into acetaldehyde, This is an intermediate here. And there's an enzyme that does this, alcohol dehydrogenase, And it's the acetaldehyde that's really providing kind of a toxicity. It's not so good. It's a more potent toxin than ethanol itself. And it causes all the deleterious effects if you imbibe too much, cause hangover, headache, elevated heart rate. It can ultimately cause liver toxicity if you um, abuse, how much ethanol you take in. And it can damage cell membranes, promote scar tissue, impair central nervous system. The acetaldehyde then can also be converted into acetate, okay, in mitochondria. So there are these steps here, ethanol to acetaldehyde to acetate. And what's happening in each of these steps is NADH is getting produced. Okay, just remember that. I think what you want to know here is, this is the deleterious thing, this acetaldehyde. And for ethanol, methanol, or ethylene glycol, these are all alcohols that can be abused. Sometimes methanol or ethylene glycol, if people cannot get access or it's restricted from them, they may also drink methanol or ethylene glycol. Has kind of a sweet taste. And in these cases, what you will see, I mean, in the case of methanol and ethylene glycol, it's these end products, formic acid, glyoxylic acid, and oxalic acid are really quite, toxic. But what all these pathways have in common is this generation of NADH. Okay, so there will be a lot of NADH that's being made. We skipped this here. And so the consequence of this is that if you have a lot of NADH, what ends up happening is that you stall the TCA cycle. Now, that's kind of fascinating here, is that in this step here, in the TCA cycle from isocitrate to alpha-ketoglutarate, you generate NADH. But if you have a lot of NADH already present, you don't have much NADH. So this will be decreased, this step. And so will the next step here. If the NADH to NAD plus ratio is too high, you will not make it around the TCA cycle. And what happens here in this step over here is that this reaction here goes from NAD plus to NADH, but this is a reversible reaction. So if NADH is high, this reaction is going to go in the opposite direction. So you won't be able to produce oxaloacetate, and this is going to decrease. And oxaloacetate is a way in which you could make more glucose. So you don't have to know all these details, but you see that the consequence of ingesting ethanol can have all these downstream effects that really is linked to this ratio of NADH to NAD plus. And if you have, TCA cycle stalls, acetyl-CoA will accumulate, right? Acetyl-CoA accumulation will make then that you have more ketone bodies. And the accumulation of ketone bodies can now cause ketosis, the drop in pH in your blood. And the lactate, because you can't go into the TCA cycle, the lactate, that might have been, if pyruvate cannot go to acetyl-CoA, you'll just make more lactate. So you're making a lot of acid, and now, it's not good. But I mean, you would really have to be abusing ethanol, at a chronic state. But I mean, just to know this kind of makes you think a little bit harder about that. Anyway, but consequences of having this, altered NADH high ratio, that can also lead to more triglyceride synthesis. It will decrease fatty acid oxidation, right? Because NADH is a product of that. If NADH is high, fatty acid oxidation is not really going to proceed. And what you'll see also in the next part, when we talk more about these lipoprotein particles, there's also, negative effects on lipoprotein particle formation. But these, I think, are just things that you be aware of. You don't need to know all the gory details, but how, these various biochemical pathways impact other pathways, that they're all very much interrelated with one another. And the more you understand that, I think the more fascinating it becomes. So at the end, I don't know. I don't know if you have that audience response business here, but in other classes where they do have it, I just kind of like to see, what's on students' preference lists. So I will ask you here, in this, how many of you, let's say, after a hard week of grad school or whatever labor may be, stressing you out, how many would go for a beer? Okay, 5 or so.
Only once when I was in.
Yeah,
It did not have alcohol. I was just raised not to drink alcohol, that's just me, but I mean, I'm drinking coffee
okay,
but not drinks or, I'm just saying my perspective.
okay,
And if it felt healthy, I would drink orange juice, but I would make it that.
okay.
That's just, my.
What about red or white wine? Who's kind of into wine? Okay, we have 1, 2, 3, 4. Wine, more the preference. Now, what about liquor? Like, okay, oh, the same beer people also like, Scotch, tequila, vodka. Okay, this has happened before. You know that you can choose multiple things. Okay, so what about cocktails? Okay, you got to go. Okay, cocktails. I think in other classes, the same ones up there. These are the real ones. They like the beer, the scotch, and the cocktails. That's what you cannot reveal when you do that audience response, because people can only choose one thing. But you see with the graduate student, I get the picture. Okay, hard seltzer.
Yeah, like how the coffee, I mean, I'm sorry, when I drink soda, like back of my teeth get jittery, but
Oh.
if I were to drink like not really soda, but like ice with like dye color to it.
Yeah.
Or like seltzer would be different because in my opinion, it wouldn't have as much sugar as like anything else.
Okay. Now, what about kombucha? Anybody? Oh, 3 we have. Coffee or tea? You want to say coffee? Coffee or tea? Okay, there's several. Bubble tea. Interesting. Yeah, bubble tea is quite popular. Okay. Water. Water's a must, right? Water's good. Sparkling or still. Juice, powertrain. Yeah. I think in general, in other classes, like let's say in the other PhD program classes or in med school, I think the most popular one, but they use thing is cocktails. That seems to be the more present one. Yeah. Yeah. Okay.
Can be dangerous, so instead of energy drinks, I think it would be better to drink coffee or tea with caffeine. That's just my opinion. That's just my opinion. Yeah, you got it. Yeah.
Okay, but for those who like work out, who drinks like those protein drinks, like core, yeah, right? I mean, it's amazing how they keep packing so much protein into like these bars, 28 grams, in this David bar, or 40, grams of protein and core protein, something or other. You see it there. It's incredible. Because I think we're supposed to eat more protein, and so suddenly protein is appearing in everything. Okay, now, at the end of my lectures, I just put, review questions, things that are mainly the key aspects of the lecture. I mean, it's a multiple choice, exam questions, and there's no trick questions. I mean, it's mainly the more important things. Okay, so that's that. Now, see, so it's recording. Want to keep your ink annotation? Well, I didn't do anything, but we'll just say keep, why not? And then we keep recording, and so we just close this and go to the next lecture, unless somebody really has an urgent need to take a break, we will, I don't say, okay, we'll go to the second one.
Oh, Caroline, this
Yes.
is Diego.
Hi, Diego.
Um, so you can change your file. It keeps recording.
Right. That's what I was going to do. So I just go to the next lecture.
Yeah,
Yeah.
right. Great. Yeah,
Okay. Great.
thanks.
Yeah. Thank you for logging on because Alika wasn't able to start the
Yeah.
recording.
She
Yeah.
needed the code. Yeah.
Right.
But
Okay. Great.
yeah, we didn't miss too much actually.
No, that's okay. It was only just a few minutes. No
Yeah.
problem.
Okay,
Okay.
great.
So membranes, part two, we focus more on cholesterol synthesis, its pharmacologic regulation, and how cholesterol is transported throughout the body. And I think this touches upon a little bit of membranes, a little bit of biomedical, drug discovery. There's genetics, there's biochemistry. I think everything is contained in here. And I like it because no matter where you are, in the healthcare activity, exercise, you can make an impact, whether you are the individual person, your health also, to be cognizant about, cholesterol levels, what you eat, how you eat when. If you do research, for drug discovery, there's so many other drug targets that still need to be explored here if you're going to go into medicine. I think that's a real crucial thing because cardiovascular disease has really, gone down with greater awareness and pharmacologic agents in order, to keep people fit. And also, in terms of if you're going to go into dentistry, I mean, this is also really important too. I mean, gum health and the microbiome in gums, et cetera, it's really crucial. And a lot of it can be influenced by, this whole process of cholesterol synthesis and regulation. So cholesterol is in cellular membranes. What you see here is just a cartoon, and it's a phospholipid bilayer shown here. So these lollipop-like structures with the polar head groups, the fatty acid tails, these are phospholipids. This is facing the extracellular space. This is facing the cytosol. And these stubby little purple structures, which are shown here, is cholesterol. And so cholesterol is also found in membranes at different proportions, depending on what membrane it is. And it really imparts this kind of curvature and stability of the membrane, that will give it more structure. There's certain areas of membranes that are called like lipid rafts that have like an increased amount of saturated fatty acids, of sphingolipids, and of cholesterol. And so they can be entry points, like for viruses into cell, or egress points for when viral particles are released from cells. And so it's important in membranes, and they're present, throughout various membranes of the body, of the cell. And so what you see here is that in cells, there's an increasing gradient of membrane cholesterol. And it's lowest in the endoplasmic reticulum, which is shown here, right? The nuclear envelope and the endoplasmic reticulum, which is shown here with these green balls on it, those are ribosomes, is continuous with one another. The nuclear envelope and the ER, the rough ER that has the ribosomes on it, are continuous. And then it goes into the smooth ER. The smooth ER is what forms vesicles, transition vesicles that can move proteins and contents of the ER from the ER to the Golgi complex, which is shown here. So as we're moving to the outside of the cell, the proportion of cholesterol within the membranes is increasing. All right, so the highest amount of cholesterol would be found at the plasma membrane, which is shown here. Also, cholesterol is a precursor of steroid hormones, vitamin D, and bile acids. And so it's important in that way, is that cholesterol, help to form steroid hormones like estrogen, glucocorticoids, testosterone, also to form vitamin D, to form bile acids, which I told you about previously. These are important for emulsifying fats that are in your gut. And so cholesterol has 27 carbons. It's shown here. And there's like a ring-like structure, like you saw in the cartoon here, this little purple stubby structure. And the two carbons, which are shown here, can vary. There can be a hydrocarbon tail of various lengths here, and also there can be an esterification at this carbon here, where fatty acids can be placed, right? So all cholesterol molecules are not identical. They can have different fatty acids, they can have a different length of hydrocarbon tail. And it's synthesized principally in the liver and also in the intestine. We don't require it in the diet. If you have a normal physiology, you won't need to take it in a dietary way. But of course we do. If you eat a lot of foods that are high in cholesterol, for example, coconut has a lot of cholesterol in it, then you'll increase your cholesterol levels that are present in the body. And there are not many ways of eliminating it. The only way to eliminate cholesterol in the body is when it gets converted into bile acids. It gets stored in the gallbladder. And when you eat something, then it gets secreted into the intestine, and only a small percent of the bile, right, that's surrounding lipids will be excreted into the feces. The remainder are reabsorbed. So only like 5% of cholesterol that's present, in bile acids can be eliminated from the body in that way. And the principal form of cholesterol that gets transported throughout the body is a cholesterol ester. So it's this cholesterol that has a fatty acid linked here. And this is the form that's primarily packaged into these particles, these lipoprotein particles that I'm going to tell you about. And cholesterol synthesis, it's a long pathway, okay? And these are just the steps. It's no need to know all the various steps, but 2 points to point out is that the rate-limiting enzyme in cholesterol synthesis is HMG-CoA reductase, all right? It's the slowest, the most regulated step in cholesterol synthesis, and it's a key drug target. For all those cholesterol-lowering drugs that you see advertisements for, statins, HMG-CoA reductase is what is being regulated. And another thing that I just kind of wanted to point out is that, the synthesis pathway, is going on in various compartments within the cell. In the cytosol, at the ER membrane, that's where HMG-CoA reductase is located. Also in the cytosol, also in the endoplasmic reticulum. So, you get this image of a pathway, synthetic pathway in a textbook, and you have this impression that these enzymes are all kind of shoulder to shoulder, funneling off their intermediates. But this is not happening like that. It's kind of bouncing out throughout the cell, which probably in part the more possibility to regulate the pathway, the synthesis in time and space. Also, another important thing, the bottom line here, is cholesterol is synthesized when ATP is abundant. It's something like 34 molecules of ATP is required to make 1 molecule of cholesterol. So even though, we ingest food that has cholesterol, it's not a fuel. No, we're not getting any energy derived from the cholesterol. It costs a lot to make it. And so you only synthesize cholesterol when ATP is abundant. And so the cholesterol synthesis is regulated, and there are various ways in which that can be done. And one is through gene expression. So cholesterol-regulated gene expression. If the intracellular level of cholesterol is high, then transcription of nearly all the genes encoding proteins in that pathway I showed you in the previous slide will be downregulated, right? There's a transcriptional way to just shut down the expression of those various genes. mRNAs that are going to encode enzymes in the cholesterol synthesis pathway. Conversely, if intracellular cholesterol level is low, transcription of cholesterol synthesis genes is going to go up. All right, that makes sense. And the transcription factor regulating many of these genes is done so by this SREBP, the sterile responsive element binding protein. Okay? And it's a very fascinating transcription factor with some unique features. So this is just summarizing what the SREBP is all about. So it's binding to the sterile regulatory element. Okay, that's a promoter element, upstream of all the genes that are involved in the enzymes that encode enzymes in the cholesterol synthesis pathway. And SREBP is a transcription factor, but it's a transmembrane protein, has a DNA binding domain, and it has also another SCAP, which is a protein and interacting domain. And SCAP, it stands for SREBP cleavage activating protein. It also is a transmembrane protein. It has a sterile sensing domain. You'll see this in the next cartoon. Yeah, but this lists it here, so you don't have to write it down. And it binds to SREBP in the endoplasmic reticulum, so that when ER is sterile in the membrane of the ER membrane, when sterols are low, then this SCAP SREBP complex is going to move to the Golgi complex. And once it's in the Golgi, there are going to be two proteases that are going to clip this complex, and now this maturation process is going to occur, so that the SREBP, which is a transmembrane protein, is now going to be released from the membrane so it can go into the nucleus and activate transcription. So it's easier to see it in pictures. So we're going to be talking about three organelle systems, three membrane systems shown here. So in the ER, right, this is the ER membrane. On the top is the cytosol, and the bottom is the lumen. And this SREBP is sitting in here. See, so it has this loop, transmembrane loop. And on one side is the transcription factor, the SREBP DNA binding domain. This is what's going to bind to the sterile responsive element at the 5' end of a gene in order to activate transcription. The other domain, SCAP, is shown here. It's a globular domain. And this globular SCAP interacting domain is going to touch this SCAP protein. And this protein has multiple transmembrane regions. And this is what's sensing how much sterol is present in the ER membrane, right? So that when sterol level is low, this SCAP and SREBP, they're going to be present in vesicles that originate from the ER. These vesicles are going to bud off, and they're going to go to the Golgi complex and fuse here, right? And when they fuse with the Golgi, its orientation is going to look like this. The DNA binding domain of SREBP is here. This is the SCAP interacting domain. Here's SCAP. And once it's in the Golgi, there's going to be a resident Golgi protein, which is a protease that's going to clip this loop. Okay? And when that loop gets clipped, this is going to raise up from the membrane, this SREBP domain. And another protease can come and clip it there. And now this transcription factor, which was tethered to the membrane, is now released. And once it's released, now it can traverse to the nucleus, go through the nuclear pore, sit on the sterile responsive element, and activate the transcription of all those various genes involved in cholesterol synthesis. Yeah. And so that's a very different kind of transcription factor, one which has a very more sophisticated, signaling system in order for its activation. So like when intracellular cholesterol is low, then this SCAP and SRBP, which would get retained in the ER when cholesterol is high. But when it's low, now something is sensed in this ER membrane, so this will vesiculate, make these vesicles that are going to go to the Golgi, fuse with the Golgi. Now there are these two clipping things that happen here. I mean, it makes sense. The first one's going to happen in the lumen of the Golgi, and the second one is going to happen outside in the cytosol, so that now this blue SREBP transcription factor can go to the nucleus and now activate transcription here. Okay, so that's transcriptional regulation. Another way in which cholesterol synthesis can be regulated is by the degradation of HMG-CoA reductase, right? The rate-limiting enzyme in cholesterol by synthesis. So if cholesterol level is high, and we don't want to use all kinds of ATP in order to mediate its synthesis, then the stability of HMG-CoA reductase is going to go down. And the way that happens is that when cholesterol is low, this HMG-CoA reductase, it's also got many different transmembrane domains here. It's correctly folded. You know, all of the transmembrane regions are in some orderly fashion for its ability to catalyze, cholesterol synthesis. But when cholesterol level is high, right, in the ER membrane, that can be more cholesterol, more sterols there. What happens is that now there's this misfolding, and this doesn't look to the cell like it's proper. And so it will get degraded. Now that's an irreversible process, right? You got to synthesize more of this in order to replenish it. So this gets degraded by, if you want to know, it's the 26th proteasome, which is a very big protease in the cytosol. But the degradation part is the important thing to know, that when you have a lot of cholesterol, we can just shut it all down, the synthesis, by degrading the rate-limiting enzyme. And as you might predict, there's also phosphorylation. Phosphorylation can also inactivate HMG-CoA reductase, right? And what is the kinase that's going to do that? It's the same one that was used for inactivating ACC. ACC, rate-limiting enzyme in fatty acid synthesis. Fatty acid synthesis uses a lot of ATP. Cholesterol synthesis uses a lot of ATP. So it's this AMP sensing kinase. When energy is low, it's going to be active and it's going to phosphorylate HMG-CoA reductase in order to shut it off. In the same way that AMP kinase phosphorylates ACC in order to shut off fatty acid synthesis. And then in the same way as we talked about for fatty acid synthesis, hormones also regulate the expression and activity of HMG-CoA reductase. Insulin, same reasoning, is that if you have insulin, more glucose inside the cell, more glycolysis, more ATP in order to fuel oxidative phosphorylation. So this will stimulate HMG-CoA reductase expression and its activity. Glucagon would do the reverse. And then the last way in which we can, regulate HMG-CoA reductase is through drug inhibition. All right, and so HMG-CoA reductase is a major target for drug discovery. So that here, New Jersey, we had many, pharmaceutical companies, Schering-Plough, Merck, Bristol Myers Squibb is also located here, Celgene, I mean, the list goes on. But there are all these companies who have their own version of a statin, which they all do the same thing. They competitively inhibit HMG-CoA reductase. But what makes these all different is that it's a different small molecule. All of them do the same thing, but they all have a different chemical structure. And that's what makes it possible for these companies to patent them, because they're different from one another, and each of them are going to have a different pharmacokinetic profile, right? How they act in one person may be different from another person, and everybody is their own individual. So some people react not so well to one statin, another one will work well for them. So that's the job of the patient as well as the doctor to really, go through this, be aware of how you're feeling and fine tune maybe the dose or which particular, medicine you're taking. And so what's really so incredible about these statins is that they're a competitive inhibitor, meaning that they are binding to the active site. But in the case of this enzyme, what they are mimicking at the active site is a transient intermediate, okay, mevidyl-CoA. So HMG-CoA reductase is converting HMG-CoA, shown here on the left, and then ultimately it forms mevulanic acid. But in order to do that, there are a number of little conniptions that go on in the meantime. HMG-CoA gets converted to mevidyl-CoA, gets converted to mevylaldehyde, and then to mevulanic acid. And so the statin is mimicking this. And the fact that it's some transient inhibitor is quite good because it's only happening on the HMG-CoA reductase. It's not competing, with another molecule that might be used by another enzyme. That's what makes this quite unique and special. And so that's why this particular drug has been very successful and has good results. But other mechanisms for lowering blood cholesterol are always being examined. One, which was found here in New Jersey at Schering-Plough and Merck, was Zetia, ezetimibe. So this is the marketed name, Zetia. Ezetimibe is kind of the chemical name, not the chemical formulation, but some other classification name, ezetimibe. And its mechanism of action is that it acts at the small brush border of the intestine, and it's blocking the absorption of cholesterol, right? So that even if you ingest a lot, you could block it from getting, into your blood. And it doesn't block the absorption of fat-soluble vitamins or triglycerides. And so this is a big advantage, that it doesn't enter to the bloodstream, and for that reason, it has fewer systemic effects, because a lot of drugs like statins could get into the liver. Sometimes they can cause liver toxicity in certain individuals, and the molecule can get transformed, and then go in the circulation. But this particular drug, Zetia, does not work in that way. And so what's popular amongst, pharmaceutical companies and for drug discovery is to combine drugs together. And so there's a version where it's called Vytorin. And what it is, it's this ezetimibe, which blocks cholesterol absorption, with a statin, right? And so it's doing two things. It is that it's blocking the cholesterol absorption, but it's also now blocking HMG-CoA reductase. And so what's good about that is that when you combine the two of them together, what you'll see in this figure here is that on this y-axis, this is the change in the bad cholesterol, which I'll tell you about in a moment. But this is what they measure when they do blood work. They want to see how much of this LDL cholesterol, which is the principal particle that's ferrying cholesterol esters around, how much that's going down. So if you take Zetia, okay, that also has atorvastatin with it at 10 milligrams, you could reduce your blood cholesterol by 53%. Now, if you took atorvastatin only alone, in order to get to that 53% reduction, you'd have to take 8 times more atorvastatin. All right, so that's a real benefit, because the higher dose you take, more potential side effects you might have. Like a lot of people have muscle aching, muscle weakness. I think that's the principal one, really, with these statins that many people experience. There are other things, but that one. So by combining, for my cousin, for example, she was taking Lipitor and was really having problems, like in the morning, getting up and you feel like your legs are going to collapse with kind of a tightness and pain. But then she switched to this, It's called Vytorin, where it's a mixture of the cholesterol uptake inhibitor and a statin, and that just, reduced the cholesterol and also reduced this kind of side effects that she was having. Okay, so then for this particular part, there's also a set of review questions if you would like to just, review the. This next part has to do with how does the cholesterol get transported throughout the body? And this presents yet another opportunity for pharmacologic intervention in order to regulate cholesterol burden in the body. And so what's at the center of the transport are these lipoprotein particles, which are shown here. So it's just a spherical particles, and what they're surrounded by is a monolayer of phospholipids. So the purple is cholesterol, the yellow are phospholipids. So the head groups are sticking out toward the aqueous, toward the blood. Fatty acid tails are pointing in the center. And in the center is where the cholesterol esters and triglycerides are packed. And what distinguishes the lipoprotein particles one from the other is the protein, which is shown here as this long snaky thing on the outside. The protein is the signature of various particles that determines one from the other. So on LDL cholesterol, it's ApoB100. In the case of HDL, which is a smaller particle, you'll see in the next figure, it's ApoA1. All right, so the protein is what's characterizing each of the particles, one from the other, as well as some other properties. And so this is just showing you just the general classes of lipoprotein particles in the kind of terminology. It's kind of silly, but it makes it kind of clear for the eye, is that the bad cholesterols are shown here toward the left-hand side. Chylomicrons, which I told you about just briefly. This is what's responding to what you're eating. It's packaging triglycerides. That's its main purpose, is to transport triglycerides. Also considered not so healthy, VLDL. I'll tell you what all these initials stand for in a moment. IDL, LDL, and only one of these particles is considered good, HDL. And they're bad because they can increase your risk for atherosclerosis, the fancy name for saying narrowing of the arteries, throughout the body, is that if you start to occlude, the vasculature, now this can now increase your risk for heart attack or stroke. And so what you see here is the composition of the lipoprotein particles. So here's chylomicrons. So in this blue that they show here, this is mainly triglycerides. The maroon color, this cholesterol esters, phospholipids is the green, protein, purple. When you now look at very low density lipoprotein particle, VLDL, this will be all written out in the next slide. It has a good amount of triglycerides. All the others really have less. Cholesterol esters is the maroon. When we look at the different pillars here, the maroon cholesterol esters is greatest in LDL, right? And cholesterol, the thinking is that cholesterol is the culprit for causing, plaques. Maybe you heard that, atherosclerotic plaques. This is what's occluding and narrowing arteries. So that's why LDL cholesterol is really considered, a problem, because this is how cholesterol esters are getting out through the body. And when we look at what, has the most of something or other, HDL, most protein, really more minimal in terms of, cholesterol esters and triglycerides. And so this just, gives you a rundown of what the purpose of these are. So the function of lipoprotein particles is to transport triglycerides and cholesterol via the bloodstream to all the different tissues in our body. And these are the classes. Chylomicrons Okay, this is principally for triglycerides. Very low density lipoprotein particles, VLDLs. IDLs is intermediate. It's a very transient intermediate. Don't talk about that. LDL, low density lipoprotein particles. And LP little a. I mention this here because in terms of genetic determinants, for cholesterol, disequilibrium, like increased levels of cholesterol, the tendency toward having, atherosclerotic plaques, there are not that many, genetic influencers for that. Not like cancer. There are many different genes that could cause cancer or diabetes. Maybe there's only a handful for cardiovascular disease. And one is this LP little a. It's a derivative of LDA with an additional protein on it, apo little a. And if you have a mutation in this apo little a, you can significantly increase the risk for heart attack and stroke. Others can be changes in HMG-CoA reductase, for example. Or you'll see, maybe I mention it later, in the LDL receptor. There's all these particles as they're going through the bloodstream, how they get into cells, right? They have to bind to a receptor on the cell surface that takes up the particle. And so the receptor that binds to LDLs and also to chylomicrons is what can also have variants, genetic mutations that are going to lean you more toward cardiovascular disease. And these proteins on the surface, right, that characterize the various lipoprotein particles, why they're important? They're important for the assembly of the particle within the cells that make them, and also for the secretion of those lipoprotein particles from the cell. So that's why the proteins, they are kind of the mediators of that. And also the protein is important for activating the lipases that are going to break down the triglycerides that are inside the particles. And that's why the proteins are important on the outside of the lipoprotein particles. Here, this is just showing, the relative sizes here. So there's this chylomicrons are the largest, and they're the least dense. And really, so this one has a bit of a mistake from the NIH, but the chylomicron should be the least dense, so they shouldn't be below the VLDL. They should be, higher density goes down. So the green should be above the VLDL, but never mind, it's a minor thing. But all of these ones that are called bad are in this upper region here. LDL, IDL, VLDL, chylomicrons. Only the ones that are considered good, HDL, are more dense. And the reason why they're considered good is they can actually take cholesterol out of a membrane, even sort of trim down the plaque in an artery. And so it's believed that HDL is good. I mean, the evidence may not be as solid as it is for the pro-atherogenic bad, but HDL is considered to be involved in reverse cholesterol transport, that it can take it out from a cell, take it back to the liver so that the cholesterol can be recycled. And so here, this is quite an astounding visual take, is that on the left-hand side here, you take a blood sample, you spin down all the cells, and you look at the plasma, and it's nearly transparent. After, a fat-rich meal, there used to be a Burger King downstairs in the cafeteria, so I would say you visit the neighborhood Burger King, and then you do the blood sample, like maybe an hour afterwards, you spin out all the cells, you see this kind of opacity here. Those are the chylomicrons. In that period of time, they've packaged up the fat and released it now into the bloodstream so that it can be taken to the adipose tissue throughout the body. So that's, I think, a quite stunning representation about what's going on in the in the biogenesis of these particles. And so, the clinical consequences of having this elevated ApoB, okay, I should have pointed this out, is that in all of these pro-atherogenic, what's considered bad lipoprotein particles, they all have ApoB 100. So a lot of times if you or your parents or your friends, go to a cardiologist, nowadays, not only are they measuring your total cholesterol, your LDL cholesterol, they may also measure, your ApoB 100 as well, just to see, how much of this, ApoB 100 containing lipoprotein particles you may have. Also, more common these days is measuring your triglyceride level as well. But this is what is the clinical outcome of this, is that here is the heart, this is the left ventricle here. And so we just close in on this particular region that's boxed here. And so what you see that, in a healthy artery, blood will flow here. But if there is this plaque buildup, right, and what's in the plaques? I mean, it's composed of cholesterol, other lipids. It can also be calcium, waste products, and calcium can form here. And what ends up happening is that it's beginning to occlude blood flow in the artery. And if this becomes, completely occluded, you will have a heart attack, and that will be felt as pain. And oftentimes what happens is that it may be blocked, but then it could be that the pressure becomes so high that part of the plaque, breaks down, and now the blood flow is restarted again. And it's thought that oftentimes that reperfusion, not only is the blockage bad, but also this burst of blood that goes through and breaks through this artery can cause a kind of oxidative damage, that event in itself can be damaging as well. And so in the case of stroke, here what it is, it may be a artery, carotid artery in the neck, and then there will be also this kind of blockage that occurs here, preventing blood flow to the brain, right? And this is what's causing stroke. So you really want to, try to minimize, with age and also just your background, genetic background, and also your diet, exercise, et cetera, can influence how much of this buildup is present in the arteries. So just, picturesque notion of what's going on is that here, when it's perfectly open, not occluded, this is an artery where the blood is flowing, and there are different layers, of the muscle. The one closest to the blood is called the intima. And eventually there will be smooth muscle, okay, surrounding that. And so early on, if there is this cholesterol buildup, it may not occlude the artery here. It happens beneath the intima, like in this region here. And then as it builds up, then it will begin to narrow this artery here, shown here. But this cap that's here can become more vulnerable. They're calling it a vulnerable plaque, more thinned. And so now this will be released out into this, artery. And then it will rupture this plaque. And then this formation here, this thrombus, could harden. And now it really will become quite narrow, as shown here. And so one thing, it's not only cholesterol that's the issue and this narrowing and this plaque formation, but there's also this immune response that occurs. And this is a real eye opener here, is that the clot that's forming here, so there'll be LDL particles, right? They're accumulating out in the bloodstream. And then they can enter in through this intima, right? This layer of cells here. They go underneath, near where the smooth muscle is. And these particles will change. into something which is called oxidation. And it's basically damaging them. And this oxidized LDL will be seen by the immune system as something foreign. And so macrophages will come in and then try to engulf the LDL that's here. And they keep, consuming this, and they form these foam cells. Okay, and I'll show you in the next picture examples of, what these foam cells will actually do in the body. And so they can then release a lot of these contents, dead foam cells, right? They die. And now this is what's exacerbating, this whole process, which is shown here, underneath here, as this atherosclerotic plaque forms. But so the foam cells form these things that can be called xanthomas, and like cutaneous ones could be around the eye, eyelid, extensors, maybe around the elbow, knee. In hair, it can also affect capillaries in the eye at the back of the retina. Normally, you would see this whole network system red, blood. That's the way it should be. But here it's looking milky like this, and this is coming from a lot of, it can be triglycerides, cholesterol, which is accumulating here, even the creases of the palm. Cholesterol, gallstones, in most cases, gallstones are caused by this accumulation of cholesterol, which can be quite small, discrete little pearlettes, or really quite large, boulders, which is shown here. So, the outcomes of having, this occurring, it's not only your heart, it's, all throughout your body and the function of your eye, because this can lead to blindness, this lipemia retinalis. And so just to tell you about how do these things circulate throughout the body, these particles are not static, right? They are getting produced in the cell. In the case of chylomicrons, it's in the intestine. They get released. And so what is their life story, throughout the body? So chylomicrons are assembled in the intestine, and their signature protein on the outside is ApoB48. And so they're released into the lymph. And when they're circulating through the body, the chylomicrons will get two proteins from HDL, right? So the HDL molecule is providing these two proteins, ApoC2 and ApoE, onto the chylomicrons. And the whole purpose of that is that this ApoC2, okay, will trigger now the activation of this lipoprotein lipase. Okay, lipase, it's going to cleave triglycerides on lipoproteins. That's why it gets its name. So what's happening here is that this is the chylomicron, this yellow circle, these little tuning fork type things. This is triglyceride. On the surface is this ApoC2 that it got from HDL. And so that is going to bind to the lipoprotein lipase, this lollipop type thing here. And that's going to trigger now the hydrolysis of triglycerides that are stored inside the chylomicron. That LPL is going to hydrolyze the triglycerides into glycerol and free fatty acids. The free fatty acids are going to get taken up by that tissue. It could be the kidney, the pancreas, ovary, all the tissues of your body that would, be subject to contact with the chylomicrons. Take up fatty acids, which is a good thing because you need it for making ATP. One molecule, palmitic acid, gives you a lot of ATP. The glycerol will go back, to the liver, and it can be used for glycolysis, gluconeogenesis, lipid synthesis. So at a certain level, you need the chylomicrons, but you don't want an overabundance because that could then tilt you on the side of, disease-oriented state. And so once the chylomicrons have been, chewed up by the lipase with the triglycerides, they form these remnants, and they transfer this protein back to HDL. Okay, you needn't memorize all this, but just know that these chylomicrons, they get born, they get proteins, they change throughout your circulation, they deliver triglycerides, and then they go back to they don't go back, they go to the liver. They start in the intestine and they end up in the liver. and they get into the liver because they bind to a receptor on the liver, and it's called the LDL receptor. It binds chylomicrons and also LDL. Now, what about those other particles? They all begin in the liver and go back to the liver. So VLDL, the very low density lipoprotein, it has the ApoB100, right? ApoB100 is the one that's kind of a sign for those particles that could be risk factors. You kind of want to keep the level low. And as it's circulating, leaves the liver, circulating throughout the blood, it acquires ApoC2 and ApoE from HDL. Same thing with the chylomicrons, for the very same purpose, so that whatever triglycerides that are present in VLDL, it'll get delivered to non-hepatic, non-liver tissue. Yeah? Deliver triglycerides there. And now ApoC2 on the VLDL gets transferred back to LDL. And just know, ultimately, VLDL gives rise to LDL. The VLDL is going to give triglycerides to various tissues. It's going to get processed a little bit. It'll form LDL. And then it can also go to other tissues throughout the body, deliver cholesterol esters. Eventually, they'll go back to the liver. So there's a long path that these particles take all throughout our body in order to deliver their contents, to our organs. And so, as I said before, HDL, it's considered a good cholesterol because it can operate in the reverse. It gets synthesized in the liver and intestine. It's ApoA1 rich. It accepts free cholesterol from peripheral cells and returns it to the liver. So it can actively take up cholesterol from cell membranes, from other particles, and remove, let's say, that oxidized cholesterol that's going to trigger the immune system. Skip this. The LDL receptor. So this is a real key factor. Another drug target, it turns out. And it's the major regulator of plasma cholesterol levels because it mediates the uptake of the particles into the cell by a process which is called receptor-mediated endocytosis. So it's sort of interesting because the people who discovered this, their name is Joe Goldstein and Mike Brown. They are the ones who came up with this hypothesis that cholesterol is a paradigm, dysregulated cholesterol regulations paradigm for heart disease. And they define this whole receptor-mediated endocytosis pathway that I'll describe for you in a bit. But they are the same ones who also discovered the SREBP and SCAP. So they have a long storied career of this kind of research. At the very basic, I think one of them is a PhD and one has an MD, but they team up together in order to make these discoveries. And so this LDL receptor is interesting because it is a single transmembrane protein present on lots of different cells. And on the extracellular side, it can bind to ApoB100, okay, which is on all those atherogenic LDL particles, and ApoE, which is on chylomicrons, has a transmembrane domain and has a cytosolic domain. And this is what is mediating the receptor-mediated endocytosis. And so that process is just shown here, but we're going to just zoom in just so I tell you just some general features. It's kind of cool. So you have this LDL receptor, this green monopoly box here, house, and it's going to bind to the ApoB100. Okay, this little beak here, going to bind the monopoly house. And when this comes into contact, what happens is that the membrane here of the cell is going to form what's called a clathrin-coated pit. Clathrin, it looks kind of like a chicken wire, hexagonal chicken wire that's going to deform the plasma membrane, and it's going to surround the LDL particle and take it up to form this coated vesicle. That coated vesicle is going to go into the cell and it's going to fuse with another organelle called the endosome. And in the endosome, the pH is lower. It's like 6. Most of the cell physiological, it's like 7.4. This is more like 6. And at that pH, now the receptor and the ApoB100, these are going to dissociate. And the receptor is going to pinch off into a vesicle and get recycled to the plasma membrane to do the job all over again. So this is this receptor-mediated endocytosis. And so when it's in the endosome now, this particle, LDL particle, is going to fuse with the lysosome. And the lysosome is this organelle where the pH is even lower. It's like, 2 or 4. And it will just break down the LDL particle, break down the membranes. There are all kinds of peptidases, non-specific proteases that are going to just break it down into constituent amino acids, fatty acids, cholesterol. And now those can either be stored in fat droplets, lipid droplets, or it can be used for cell membranes or steroid hormones or making bile acids. And so it's important. We need this. And the cell can sense then how much cholesterol is available. But if that LDL receptor is not working, let's say there's some defect in it, and the LDL particles just remain out here in the cell, outside the cell, those particles then can go on to form these atherosclerotic plaques, right? Because it's not being taken up as it should be. On top of that, because it's not getting taken up by the cell doesn't know that cholesterol is available. So it will keep making cholesterol through that HMG-CoA reductase pathway. So you're making more cholesterol, you're leaving it more on the outside, and that's where really trouble really occurs. So mutations in this LDL receptor can really be a problem. So that if you have a certain mutation, even as a child, you could be having heart attack, because you cannot take up these lipoprotein particles into the cell, and they just stay out there and they do a lot of damage to your arteries. But there are ways still to try and treat this. And so this is like the newest thing that came out. There are these antibodies against this protein called PCSK9. It's got a long title that is quite different from what this function is, but it's an immunotherapy for lowering LDL cholesterol. And what it can do is you can get this when the statins or ezetimibe don't work for you. Either you can't tolerate it, or you take high doses and it still doesn't lower it. And so this can now be given to individuals, but you must have a normal LDL receptor. That's the side for this that's kind of restrictive. And so that came on the market. It was in 2015. It didn't take too long, between the discovery point and actually getting a drug. Discovery for a lot of these, targets like this come from pedigrees, from genetic pedigrees, from families where they didn't have any heart disease in their family because they have a mutation in the PCSK9 that it doesn't function. And so therefore, that's a great drug target, because if you can knock it out and you don't have a functional, version of that, then you could use a drug to inhibit it. And what it does, I don't know if you can tolerate more information like this, but is that the PCSK9 is a particular protein, and in its normal function, what it does is to bind to the LDL receptor, not where LDL binds, but to some other portion of the receptor. And what it does is to now stimulate its endocytosis. So LDL is not being bound to this, but it's still, the receptor is going inside the cell. Endosomes are fusing with lysosomes, and it's getting degraded. So basically, you're taking away good LDL receptor that could be doing its job. And so now what ends up happening is you have less LDL brought into the cell. So if you can make a drug that inactivates this PCSK9, like takes it out of commission, then the LDL receptor that you have in your body can still do its work, right? And bring the cholesterol in. So the benefit here is that you increase the amount of the LDL receptor on your cell surface, increase particle uptake, recycling of this receptor so it can do its job, and reduce LDL cholesterol in the blood. And so the new thing recently is that now they found not an injectionable, like if it's an immunotherapy, that means you have to inject, let's say, an antibody-like molecule so that you can inactivate this, but they now have an oral version that should be available next year or the year after. And so that's a big game changer, because if it's an oral pill, it will be cheaper, because to take the immunotherapy, it's like $30,000 a year. Okay, so then that's it. I think there's probably some, review questions at the very end. If you have any questions, you can ask me now, or you can email your question to me. And so then that's it. Enjoy the evening, stay warm. Diego, tell me, how am I supposed to close this down so the recording gets
Yeah,
saved?
let me stop the recording.
Okay, yeah, thanks.